Hello to our friends joining us via recording. We're starting lesson number 3 today, but before we dive into lesson number 3, we are going to do a little bit of review to help us out with the stuff from previous lessons. So we've done a little bit of time in our groups today to figure out how to correct a couple of statements that review stuff from the previous lesson. We had the 2 groups. So my friends who were in group number 1, I want you guys to help me out with your T tool that's above the screen or with that little pencil. Can we go through, let's mark first with the pencil. What are all the things that we needed to change on this one? Can you put like a mark or an X or something on all the things that you guys changed when you were in a group? The nice thing about putting a pencil mark on there is everyone can do it and we won't get on top of each other. Awesome. Any other marks you want to add, or are we feeling good? Is that all the things we changed? I think we hit most of the important ones there. All right, so let's work together. I'm going to use the chat. So help me out in the chat. The first thing that our group identified needed to change was the idea that diffusion is an active transport process. What did we need to change that first part to? Diffusion is not active transport. What is diffusion? Yeah, diffusion is a passive transport process. And when I say that word passive, what did that mean? If it's not active, if it's passive transport, what does that mean about it? Exactly, yeah. Several of us in the chat are saying that there's no energy required. Absolutely, no energy required. Okay, so the first thing that we had a problem with in this statement was it said that diffusion is active transport. If diffusion was active transport, it would take energy. It doesn't take energy, it's passive transport. That's perfect. Second thing that we underlined is the word water. When I talk about diffusion, it's not water that's moving. What was the special name of the things that diffusion moves? Do you remember what that word was for what diffusion moves? Yeah, so it's those things called solutes. Solutes are, in easy person words, the dissolved things that are in a solution. When we talk about diffusion, it's the things that are dissolved in the water that actually moved. Hey, if I wanted to talk about the movement of water, what is the name of the process that water moves in? What's that process called? Yeah, so if we actually wanted a statement to be about water, we would have to change this whole thing to say osmosis. So if we were actually talking about the movement of water, we would change our first word here from diffusion, change it into osmosis. Hey, but remind me, osmosis, is that an active or a passive transport process? If we're doing osmosis, the movement of water, yes, that is also still a passive transport process. So if we wanted to talk about water moving, we would still have to fix this to say that it's passive transport, and we would change our first word here to osmosis. But we're not talking about osmosis, right? We're doing diffusion. So far we have diffusion is a passive transport process that moves solutes, the dissolved things. We underlined to fix low concentration to high concentration. As it's written, let me show you what the slope would look like. We're moving things from low concentration to high concentration. In other words, we're going uphill. And we're going to do an example in lesson number 4 about you pushing rocks up a hill. If we go from low concentration to high concentration, you're pushing a rock up the hill. That's going to take energy. We just said that diffusion is a passive transport process, so no energy required. So we need to flip-flop these, right? We need to say that instead of going up the hill, we're going down the hill. So we start where there's a high concentration, where I have a lot of those dissolved things, and I'm taking them to a place where I have a low concentration, where there's not as many dissolved things. That's the only way you can do passive transport, where we go from there's a lot of something to where there's a little of something. That's the only way passive transport works. Okay, the last thing that we underlined in this statement is what we end up with at the end of diffusion. My statement here says we got two hypertonic solutions. The hypertonic word is the problem here, right? What word should we put in this space here? Hypertonic's wrong. What should be in that space? Exactly. Yep, several of us are saying the correct word that should be there is isotonic. And remind me, isotonic, what does that actually mean? Isotonic. Yeah, equal, the same. So we're going to end up with the same concentrations. The same concentrations. Remember, I gave you that statement, right? During our class, we said that the goal of diffusion and osmosis is to make isotonic solutions. It's to make two solutions that are the same concentration. So we're always going to move things from where it's high to where it's low, from where it's high to where it's low. We're going to do that forever until we get to the same concentration. And once we get there, then we're done. The process of diffusion and osmosis is done. Let's review though, this word hypertonic. What did the word hypertonic mean? If I have a solution that is hypertonic, yeah, it means I have a higher or more of a concentration, right? Higher concentration. What's that word that means low concentration? What's the opposite? You can just give me the beginning, not the whole word. Yeah, so if I want to talk about a lower concentration, the lower concentration is hypotonic. So lower concentration. We're going to have less of those dissolved things. So when I talk about, in my statement here, ending up with two hypertonic solutions, technically this isn't even actually possible. For me to say that I have a hypertonic solution, it means that somebody is higher, which means that somebody else is lower. So besides just not being correct, we don't end up with hypertonic solutions. We would also have to have something to compare it to, because remember, these tonic words are comparison words. So I can only ever have two solutions that are both isotonic. Otherwise, I have one solution that's hypertonic and one solution that's hypotonic. So let's review what this statement should say. This statement should say, let me get my pointer out here, diffusion is a passive transport process that moves solutes from areas of high concentration to areas of low concentration. When the process is complete, two isotonic solutions are generated. Okay, so that's our first reminder, how the process of diffusion works. Second statement that you guys worked on, I'm gonna go to group 2. Somebody in group 2, grab that pencil. Let's underline like we did before. What were some of the things that we changed? We'll probably have to make some columns, right? We'll fix this. Yeah, so we honed in on the most important thing, right? When we talk about the recipe or what we have inside cytosol and our extracellular fluids, it is not the same, right? This word the same or equal, that's not correct. These two fluids are different. So the recipes are different. I know I saw in one of my groups, we did a good thing for reviewing. We made a couple of lists. So let's do our lists together. One column is cytosol. The other one is, we're going to generally say extracellular fluid. Although we do have a little bit of differences, right, between the different extracellular fluids. When I'm talking about cytosol, let's start here on the left. When I talk about cytosol, which of these things that I see on my list do I have a lot of in cytosol? Which of these is there a lot of in cytosol? Yeah, so easiest way to do it is the K, right? K plus, K plus. That's my potassium, K plus. Got a lot of that inside the cytosol. By the way, this cytosol stuff, where is this fluid found? Where do I find cytosol? There's actually only one place in the body. Yeah, it's my one intracellular fluid, right? Cytosol is the fluid inside a cell, Because then we've got several over on the other side, several extracellular fluids that are outside the cell. Multiple different types. But remember, there's just one kind of fluid that's inside the cell, and that one kind of fluid is called cytosol. So one of the things that cytosol has a whole lot of in it is potassium. There are a couple other things that we talked about that cytosol has a lot of. Can anyone think of a couple other things we would add to this list for cytosol? What other things might we add to this list? Okay, lots of us are honing in on proteins, absolutely. Lots of proteins, and then the other thing, like we're seeing now in the chat, is glucose. Hey, remind me, why does a cell want to have a lot of glucose in it? What's the function of glucose? Yeah, so we're talking about energy, right? Glucose is what a cell uses to build energy. I kind of like to think of glucose as a cell's favorite food. Because glucose is the easiest way for a cell to make energy. So glucose is the cell's favorite food. We can make energy from it really fast, really easily. When we talk about proteins, there's a whole lot of different jobs for proteins, right? So you read about the variety of different jobs. That's everything, we'll list some of them here. That's everything from things like structure, giving a cell its shape. That's things like functioning as an enzyme to help a cell do something. It helps it to communicate. That word, well, let's see if it'll fit. Yeah, it kind of fits. Okay. Lots of different jobs that proteins do for a cell. So when we talk about the recipe for intracellular fluid, we've got that potassium, we've got those proteins, we've got the glucose. That's what we have inside the cell. Outside the cell then, we've got a lot of what we had left over, right? So the sodium, Na+, that's sodium. Yeah, we also have that Cl-, that's my chloride. And I had one type of fluid. Yeah, one of my types of fluid had protein. Do we remember which of those extracellular fluids had proteins inside of it? Yeah, that was found only in plasma. I don't have protein, so let's make a note here. No protein in interstitial fluid. Let's see if I can spell that. Yep, interstitial fluid, right there. Remember when we were talking about the recipes, that protein level is the important difference between plasma compared to interstitial fluid. Both of them have a lot of sodium and chloride. Plasma also has a lot of proteins, and those were things like your immunoglobulins, your antibodies, and things like clotting proteins, only in plasma, not in interstitial fluid. So I know this wasn't lesson 3 stuff, but both of these statements that we just corrected together and the lists that we've made, this is what I like to say is a wink, wink, nudge, nudge. These are important ideas. So we took some class time to make sure we understand the difference between diffusion and osmosis and the way that diffusion works. We took some time to compare our recipes for the types of fluid. So make sure you're familiar with those things like we just worked through today. I can promise you some test questions about those things. So thanks for fixing those statements for me. Hopefully we learned a little something. Lesson number 3 is talking about the role of the plasma membrane, or how the structure of the plasma membrane influences the way things can or can't get into the cell. So you've got 2 pictures here. The picture that I see over here on the left shows us what it's like when a cell is normal. So if you remember from our office hours, we talked about salty banana as the cell, right? So this is the banana. So we've got a bunch of potassium inside our cell, like we just said. And we took that banana with its potassium inside and we sprinkled some salt on it, sodium chloride. So I've got sodium and chloride outside. I've got potassium on the inside. Because I have different concentrations of potassium and sodium and chloride inside versus outside, there's a special word to describe what's going on with this cell. It actually has a different charge on the inside compared to the outside as well. What's the word when a cell is different on the inside compared to the outside? What would I say that that cell is if we're different inside compared to outside? Yeah, so a couple of us are chiming in. This is a really important word for us to know. A cell is polarized if it's different on the inside compared to the outside. Polarized. So when a cell is polarized, it has a bunch of potassium inside and a bunch of sodium and chloride on the outside. Now, something that often trips students up, so I want us to take a moment to think about this. A polarized cell, that's a good thing. You want your cells to be polarized because when we start talking about things like muscle cells and the way that they're going to do muscle contraction, or when we start talking about neurons, the way that they're going to send messages to each other, those cells have to be polarized. It has to be different inside compared to outside. Otherwise, those cells can't do their jobs. So polarized is a good thing. We want our cells to have a different stuff going on inside compared to outside. If the process of diffusion got its way, if it was able to just do what it wanted to do, so take everything from high concentration to low concentration, what we would see happening is this image over here on the right. So we'd start with a ton of potassium inside. About half of that potassium would go outside the cell. So see how I've got some potassium on the outside now? Because I'm going from high concentration, what was high inside, low concentration on the outside. And the opposite would be true too. Some of this sodium and chloride that we had so much of outside would go inside as well. What we see over here is a cell that's polarized, that's happy and healthy and can do its job. The cell we see on the right, we would say is depolarized. It's lost its polarity, or it's lost the difference inside versus outside. And this cell is not happy. This cell is not going to be able to do its job. So polarized cell on the left, depolarized cell on the right. Miriam has a question. Go for it.
Sorry if it's not related to lesson 3, but it's something related to polarized and stuff going in and out of the cell. I don't remember where I read that there is a limit of things that will go into the cell. For instance, if we put the cell in a hypotonic solution, the cell will allow things go out but not going in to so I'm just kind of confused because in this case the cell allowed the sodium chloride to be inside. I understand that the potassium will go out to have that ionic or switch of ions and cations, but just don't understand that part. What is the rule of for the cell to allow things in? Is there a limit, or is there a certain stuff that can be through the membrane?
Yeah, so your question is actually a perfect question for our discussion here in lesson number 3. So when we're looking at our image on the right over here compared to the image on the left, we saw that a bunch of things moved into the cell. The sodium and the chloride moved in, and the potassium moved out. But what we need to realize is that cells don't look like this normally. Like you said, we don't normally let a bunch of sodium and chloride into the cell. Normally, we keep that stuff on the outside. And actually, to be fair, we don't normally let a lot of potassium out of the cell either. We keep that stuff inside. So our goal in talking about chemistry in this lesson, lesson number 3, is to try to explain, kind of to address the question that you asked. Is there a limit, or what stops this from happening? And what stops this from happening is actually plasma membrane. So the chemistry or the structure of the plasma membrane makes it so that I can't get a lot of sodium and chloride into the cell, and I can't get a lot of potassium out of the cell, except when I really want to, on purpose, get it in and out of the cell. If I'm going to purposely get it in or out of the cell, I actually have to make a special protein channel for it, because as you're reading about in lesson number 3, these structures here called phospholipids, they're really particular about what they let in and out. And normally they're telling sodium and chloride, you're not coming in. And normally my potassiums, they're telling it, you're not going out. You're staying inside. So I'm not sure if I fully addressed your question, but I bet by the end of tomorrow, when we've covered everything from lesson number 3, we will have addressed it. But the short answer is that absolutely, there's a limit to what can go in and out. And a lot of that limit is just based on the fact that the plasma membrane says, no, you're not moving. So hopefully that clears it up a little bit. This is never going to happen in your body unless your cell is dead. If your cell is dead, it will depolarize, and it'll be the same on the inside and the outside. If we're the same on the inside and the outside, we can't do our job. We need to be polarized. We need to be different. So the plasma membrane helps to keep us different. Help me out in the chat. Thumbs up, thumbs down. What questions do we have right now? Okay, got some thumbs up. Perfect. Okay. So the purpose of looking at these two pictures is to help us realize that there's a reason that we need to care about the chemistry of the plasma membrane to make sure this doesn't happen. This is dead cells. This is bad news. So we're now going to dive into our chemistry of how we keep a cell polarized, how we make this happen. So let's talk about the plasma membrane. The plasma membrane is known as a couple different names, but the name we're going to focus on calling it is the phospholipid bilayer. And in your guided lesson, it breaks down this name, phospholipid bilayer, for you. Emphasis with this bilayer word that there are 2 layers of phospholipids. So when I look at a very simplified cell, your cell is never going to be just this simple. There's always going to be proteins and other stuff. But when I look at a simplified cell, I can see on the outside here 1 row of phospholipids, and I can see in the middle another row of phospholipids. Over here on the right, you can see zoomed in big what a phospholipid looks like. So the phospho part of its name comes from the phosphate group up here. And the lipid part of its name comes from these 2 down here. So let me put that word lipids. Lipids. Fatty acids are the same thing. They are a type of lipids. So a phospholipid has a phosphate group in this area called its head. And then it has these lipid tails. I arrange my phospholipids in a bilayer because of their interaction with something. What did our guided lesson say made us form this phospholipid bilayer? It forms because these guys are interacting with, they're interacting with water. So the phospholipid bilayer, it forms because of water. Now, when we're looking at this picture of the cell here, I have the inside of the cell and I have the outside of the cell. Where is there water in this picture, where would I find water? We feel like it's a tricky question. Where might we find some water? It's okay to guess if it's wrong. Yep, we definitely have some water here on the outside. Outside, oh, I'm going to type water here, H2O. The water on the outside of the cell is that plasma. It's that interstitial fluid. Okay. And check this out. We also have water on the inside. What's the name of the water and the stuff that's in it, for that matter, that's inside a cell? What's the name for that fluid inside the cell? Yeah, cytosol. Cytosol. Okay, so check this out. I have 2 layers of phospholipids. These 2 layers of phospholipids, in the very middle of them, inside the cell, there's water. On the very outside of them, we also have water when we're outside the cell. But notice the parts that are touching water are always these little circles that we see right here. See how this part's touching the water out here? And this line of circles is touching the water here in the middle. The parts of a phospholipid that are touching the water are always the head parts. The heads are always right next to the water. The tails, the lipids, they are always as far away from water as possible. So when you're thinking about water inside and outside a cell, the big thing just to keep in mind, these lipid tails right here, there is never water right here. There's water all over the place outside the cell. There's water all over the place inside a cell. But in the middle of its plasma membrane, we've got this special space in here where there's no water. And the fact that there's no water in here is part of why, in relation to the question that I think Miriam asked, that's why sodium and chloride and potassium, they don't just go inside and outside as much as they want to, because this area right here where there's no water, this area says, you shall not pass. You're not going to get past me. I'm going to regulate what comes in and out. So big ideas we've talked about so far. Water inside a cell, water outside a cell. The plasma membrane, what frames in the inside and the outside of the cell, is made of these guys called phospholipids. The phospho part of its name is these heads, these circles. The lipid part of its name is this stuff here in the middle. So this is where your lesson took a turn for, depending on whether you like chemistry or not, right? We took a turn for the worst. We started to talk about chemistry. I've said it before, I'll say it again. Chemistry is not my favorite, but we'll make it through chemistry together. So now we need to talk a little bit about the chemistry of why water makes us form that bilayer. Before I do, I see Miriam's got a question. Go for it. Oh, maybe it was on accident. Go for it.
Yeah, sorry. So before we move to this slide, can we go back to the other slide?
Yep, my writing will be gone, but we can go back.
Oh,
yep.
Okay, perfect. So do we need to know the that division on that second, the right picture, which has a phosphate, and the second part is that glycerol?
So all you need to know is that in the head we have a phosphate group.
Okay.
That's all we need to know. We don't need to know the structure of a phosphate group. We should know with the phosphate group, let me pick color, it'll show up. We should know that phosphate group has a partial negative charge on it. So phosphate has a little bit negative on it, negative. So we want to know that the phosphate has a negative charge.
Okay.
And we want to know down with, remember we labeled this the lipids. We want to know down here with the lipids. This will mean more here in a moment, but lipids are non-polar. Lipids are non-polar. So I've got a head group with a phosphate that has a partial negative charge, and I've got lipid tails that I see down here that are considered non-polar. But I'm not going to ask you to draw me the shape of the head. I'm not going to ask you to draw me the shape of the tails or to identify the glycerol versus the phosphate. I just want you to know that a phospholipid has a phosphate in its head and it has those lipid tails. That's what we need to know.
Okay, thank you.
Mm-hmm. All right, so let's talk chemistry. When we talk about everything in the chemical world, we are talking about elements. So periodic table has all of the elements, the names of things on them. So for example, I've got an O up here. O stands for oxygen. Down here, I've got an H. H stands for hydrogen. We are looking at the chemical elements that I find inside water. Remember water, so you probably know this from a chemistry class. Water's chemical formula is H2O, meaning I have 2 hydrogens and 1 oxygen. That's what H2O means. When I look at each of these elements by themselves, there are a couple of different areas that we see. So the first area here in the middle that's labeled with these letters for us, that's the part of an element that's known as the nucleus. The nucleus, just like we use that word nucleus for a cell. Now let's do a little bit of lab review here. When we talk about the nucleus of a cell, what's inside the nucleus of a cell? What do I have inside there when it's a cell? Yeah, so a couple of us are chiming in. That's where we find the genetic information of the cell. So that's where we find our DNA. When normally it's loose, which is, like Miriam mentioned, that's the form called chromatin. So normally our DNA is loose and it's chilling out in the nucleus of the cell. Well, we've zoomed way in. Now we're not talking about the nucleus of the cell. Now we're talking about the nucleus of an atom. Can anyone help me out in the chat? Do we know what we find in the nucleus of an atom? This really small area in the middle of my element. What do you find in the nucleus of an atom? Yeah, perfect. So a couple of us are chiming in here for us. When we talk about the nucleus of an atom, we're finding things called protons and things called neutrons. Absolutely. Protons and neutrons live in the middle of an element in that area called the nucleus. Floating around them, like we see on this picture here, or we can see them in these little dots right here, these are the electrons. Yeah, so the electrons actually form kind of like a cloud around the nucleus because electrons are constantly moving. They're going all over the place in circles. Protons and neutrons are chilling out here in the nucleus. They don't move, but those electrons move. Okay, so let's talk charges. When I talk about a proton, what kind of charge does a proton have? Yeah, protons are my positively charged ions. So they've got or element, subatomic particle. Let's be specific here, right, for our chemists. So protons have a positive charge. Electrons have what kind of charge? Electrons are, they are the negative ones. What kind of charge do neutrons have? Neutrons, can't spell them. Yeah, neutrons are neutral, so no charge, they're neutral. Okay, so we have an element like oxygen. In the middle part here, it's got its positive protons and its neutrons that are neutral, no charge. Circling around it then are the negatively charged electrons, and they're going to constantly keep circling around this nucleus because electrons have a negative charge and the protons in the nucleus have a positive charge. So keep in mind that the nucleus, the center of an atom, has a positive charge overall, and we've got negative charges that are swirling around the outside, and they keep swirling because opposites attract. That's kind of the big idea of chemistry for us to know is opposites attract. So negatively charged electrons, positively charged protons in the middle, they're constantly attracted to each other. This electron cloud, by the way, it's darker in places where we see a lot of something, and it's lighter in places where we don't see as much. So the electrons spend a lot of time really close to the nucleus, not as much time farther away. And at some point, they're never out this far because then they can't feel that positive charge from the protons. So electron clouds give you an idea of where electrons are going around in circles. When we start to form compounds, so things like H2O, the electrons that were circling around one nucleus, they might start circling around another nucleus as well. So here's one nucleus, we got our protons here. Normally I've got electrons that are going in circles like this. Well, when I form a chemical bond, my electron that was zipping around like this suddenly says, oh look, there's some positive over here. And it'll do a loop over here. And then it says, oh, there's some positive over here. And it does a loop over here. It goes back and forth around both of these nuclei. That's one way to form a bond, if I'm going back and forth equally. I could also form a bond where I spend a lot of time orbiting over here, and then I say, oh, look, there's something over here. Loop it once, go back over, circle a couple of times, 3 times. Oh, I'll go over here really briefly and go back and forth. When I talk about forming a chemical bond, any kind of chemical bond, when I put 2 atoms together, any kind of chemical bond, it's all about sharing electrons. And hey, underline, highlight, star, or make a big note for yourself, it's the electrons that I share. I do not share protons, I do not share neutrons, I only share electrons. So when I form a chemical bond, it's because 2 elements, or maybe more than 2, are sharing electrons with each other. Now, some of us have seen this in the notes outline. I can share them equally, like I see here. I know it's equal because the size of the 2 electron clouds is the same. Or I can share things unequally. See how my cloud is bigger on this side than it is on this side. Anytime I'm sharing my electrons, I have a word for a type of chemical bond that I call a sharing bond. Sharing bonds are called covalent bonds. Covalent bonds are sharing bonds. So in either of these pictures that we see here with our chemical compounds, I am sharing electrons because my clouds, the space where those electrons are going, they overlap with both of these. Overlapping clouds means we're sharing. So both of these are examples of covalent bonds. In this example, I'm sharing equally. In this example, I am not sharing equally. Can someone help me out in the chat? If I share electrons equally, what's the special name for that kind of covalent bond? If we do equal sharing, that one's called absolutely a non-polar covalent bond. Non-polar covalent bond. If we were in class together in person, I would literally ask you like every day, 5 times a day, what the word covalent meant and what the word nonpolar meant. A covalent bond is a sharing bond. A nonpolar covalent bond is a equal sharing bond. So these would be terms right here. We want to make sure we 100% understand what this name means. Nonpolar covalent bond means we're sharing electrons and we're sharing them equally. If I am not sharing my electrons equally, what do I call it when it's unequal sharing? What kind of bond is an unequal sharing bond? Yeah, now it is a polar covalent bond. Polar covalent bond. Because this polar word means unequal. Covalent still means it's a sharing bond, but again, polar means unequal sharing. Now, When we talk about chemical bonds, here's a big picture idea. Keep this in mind. Anytime we're sharing electrons, that's a strong bond. So whether it's a non-polar covalent bond or a polar covalent bond, both of these types of bonds are strong bonds. The strongest type are non-polar covalent bonds. Still pretty strong, but not quite as strong, are these polar covalent bonds. Think about this in real life. If you're in a relationship with someone, the strongest kind of relationship you can have is where both of you are doing give and take, right? I give you something, you give me a little something back. We're giving and taking back and forth equally. If somebody in the relationship is a little higher maintenance, they require those electrons a little more often, it's not quite as strong of a bond. But if there's still some give and take, then it's still decent. So equal sharing bonds, non-polar covalent bonds, these are the strongest type of bonds that we have. Unequal sharing, because we're still sharing, is a pretty strong bond, but it's not equal, so it's not quite as strong. Help me out in the chat. Thumbs up, emoji, thumbs down, questions. Where we're going next, as a heads up, is dipoles, how this relates to dipoles. Okay, getting lots of thumbs up. Excellent. I like that, Isabel. That's great. I want to have a random little dance party in the chat just because we haven't had one yet. So here's a little dance party. All right, so let's talk about then what happens with these polar covalent bonds. So say I am not sharing equally. Let's talk about what happens with that. So let me skip that. We'll come back to that. Okay. When we talk about not sharing electrons equally, here's another example for us of electron clouds with their sharing. So remember that on an electron cloud, the darker an area is, the more electrons spend time there. The lighter an area is, the less time the electrons spend there. So when I'm looking at this image of what water looks like when I form this compound, notice that there's a really dark area by the oxygen and there's a lighter area by these hydrogens. That means that the electrons are spending more time by the oxygen and they're spending less time by the hydrogen. But notice how I've got one big electron cloud here, kind of shaped like a triangle. The electrons are being shared. So oxygen is still sharing the electrons with the hydrogens here. It's just that oxygen is a little more high maintenance and it needs to have those electrons more often. So if we were to look up close and personal, here's oxygen and the two hydrogens here. These little black dots are representing electrons. These electrons are being shared between the hydrogen and the oxygen, but they spend more time orbiting around oxygen than they do around hydrogen. So it looks something like this. I do a circle and a circle around oxygen, one quick circle around hydrogen, and then do a few more circles here around oxygen, quick circle around hydrogen. I spend more time circling around oxygen than I do around the hydrogen. This is where we generate those things that you guys mentioned we wanted to talk about. Dipoles. Dipoles. This little Greek symbol here means dipoles. Dipoles, we'll make a definition for ourselves, are areas where electrons spend more or less time. Oops. There we go. Dipoles, areas where my electrons are either spending more or less time. The way that you end up seeing that then on a molecule is the other definition of dipoles. Dipoles are areas of partial positive or negative charge. This partial word is important, and I want you to keep that in mind with dipoles. Dipoles are areas that kind of look more negative, but they're not 100% negative. Because see, we talked about things like sodium or potassium that are always positive, or we talked about chloride, how that's always negative. Dipoles are not areas that are always positive, or they're not always negative. It's just that most of the time when I look at them, they have more electrons floating around them, or most of the time when I look at them, they have fewer electrons floating around them, but they're not fully negative or fully positive. So when we look at water, the dipoles in water, the area where those electrons spend more time is the negative dipole. In the case of water, that's around oxygen. The areas that are a positive dipole are where those electrons spend less time. That's down here by the hydrogens. So areas with more electrons, or more often where the electrons are, they're going to have a negative charge, partial charge, or a negative dipole. Areas where those electrons spend less time, they're going to have a more positive charge. Remind me, because it makes a difference here, the charge on electrons, were they positive or negative? What's the charge on electrons? Electrons have they have a negative charge. Absolutely, they have a negative charge. So if we have more time with electrons, here I'm going to abbreviate them, electrons, more time with electrons means it looks negative. And I have more time that the negatively charged things are by me, I look negative. Less time with those negatively charged electrons means I look slightly positive. Slightly positive. So a negative dipole means that the electrons spend more time in that place. A positive dipole means the electrons spend less time there. Thumbs up, thumbs down, questions. What do we think about dipoles right now? Ariel is cautiously thumbs up. Okay, so I've gotten a lot of at least cautiously optimistic about it. Let's then use those dipoles to talk about how we get dipoles, and then we'll talk about how they contribute to hydrogen bonds. What we're looking at right here is the periodic table of elements. Now, on exams, if I am going to ask you to do anything with elements, 100%, I will give you a periodic table. So please do not memorize it. Please don't memorize the colors. I'm not going to make you do anything like that. It's not a chemistry class. So when we look at the periodic table of elements, this one that you're looking at right here is color-coded by something called electronegativity. Electronegativity. The best way to think about electronegativity is how badly an element wants electrons, how badly it wants them. By things that are these darker blue colors over here, these blue colors are elements that really want electrons. They want to have those electrons by their nucleus as much as possible. My red colors that I see over here, red and orange, these are elements that don't really care about their electrons. They're willing to give up their electrons. They don't need them, whatever, it's all good. You can talk to your chemistry teacher about exactly why that is. It has to do with the arrangement of the electrons in their outermost orbit where the electrons are going. But what you need to know for the sake of our class is that if we are red or orange, we're less likely to need or to want those electrons by us. If we're blue, we really want those electrons by us. All of these things here in green, so the green elements, they want to share their electrons. And so they kind of want to share them equally. So if we were to pair a couple of green elements together, they would share their electrons equally, no big deal. Even if we pair some of our greens with some of the lighter blues, for example, they would share electrons equally. If we start pairing, though, some of these dark blues with a green, or definitely if we try to pair a dark blue with a red, they are not going to share their electrons equally. So electronegativity is a measure of how badly we want to have electrons by us. If you're really electronegative, you really want those electrons by you. If you're not very electronegative, then you don't care as much. You're willing to give them away. Most of the stuff that we care about in the course of our class is going to be stuff that kind of falls in the green to blue spectrum. We are mentioning, though, check it out over here, potassium and sodium. These are red. And when we talked about potassium and sodium, we talked about them with a positive charge, right? We did Na plus and K plus. That's because they like to give away that electron. So see how they're here in red? They give away that electron. So big idea is if you are very different colors, if your electronegativity is very different, you will probably form an unequal sharing bond. So let's make some notes here. Very different electronegativities. That's going to be unequal sharing. Help me out in the chat. What's the name of an unequal sharing bond? An unequal sharing bond. Yep, so the first part, the unequal part, is the polar part of its name, and the sharing part of its name is covalent. Polar covalent bond. If we have very different electronegativities, polar covalent. If we have very similar electronegativities, we're going to do equal sharing. And help me out in the chat. What's the name of equal sharing bonds? Equal sharing. Yeah, those ones are non-polar. Non-polar covalent bond. So the closer your electronegativities are, the closer we both are to wanting electrons the same amount, the better we are at sharing things equally. If we aren't equally attracted to electrons, we're probably not going to share them equally. So when we talk about something like water, we've already looked at water, so let's start by looking at this area over here. Remember that when we looked at water, the oxygens really hogged those electrons. The oxygens wanted to have electrons next to them. I'm gonna bounce back. Sorry about that. Water, dark blue with green. When I put a dark blue with a green, the green is going to get those electrons a lot less than the dark blue is. So we're going to see a polar covalent bond because we've got a dark blue paired with a green. So polar covalent bond here. Here's my dark blue, oxygen. It wants those electrons more. Here's my green, that was the hydrogen. They want the electrons less. So we generate these dipoles. That's what we see right here, these little partial positives, partial negatives. Here's a wink, wink, nudge, nudge. I want you guys to write this down on your notes packet. Every time, let's type it, every time you have a polar covalent bond, you have dipoles. That's a, like I would say, a underline, highlight, star idea. This is a must know. Every time you have a compound that has polar covalent bonds, you will always have dipoles. Dipoles are areas where there's partial positive or partial negative charge. The only way to get partial positive or partial negative charge is if we're not sharing electrons equally. So a polar covalent bond, we don't share electrons equally. If I'm not sharing them equally, I've got these dipoles, the partial positive and the partial negative. So here's why I told you guys to write this down and underline, highlight, star it. Because if I were to ask you on an exam, for example, I have a molecule that has a positive dipole and a negative dipole. Which kind of chemical bond does it have inside of it? Because I told you that there are dipoles in this compound, you would be able to tell me that they have polar covalent bonds. And the opposite would be true too. If I tell you something has a polar covalent bond, you should be able to tell me right away that we have dipoles. So this is an underline, highlight, star statement right here. Basically what it means in easy person words, if we don't share our electrons equally, that's this polar covalent stuff, then we're going to have areas of partial positive and partial negative. That's those dipoles. Unequal sharing equals areas that have partial positive or partial negative. When we talk about water with its partial positives by the hydrogens and its partial negative by the oxygen, we see these things that you guys mentioned in our word cloud called hydrogen bonds. When we talk about hydrogen bonds, I think I put it in your guided lesson. I kind of like to put the word bond in quotes. Hydrogen bonds, what might be a better way to think about them is hydrogen attractions. We don't actually share electrons. We don't actually transfer electrons. But what forms a hydrogen bond is when I have dipoles. In particular, when I have a positive dipole by hydrogen and a negative dipole by something else. So we see hydrogen bonds in water where the negative dipole of oxygen is attracted to the positive dipoles that I see in other molecules of water. This is why you can see this picture up here. You can take some drops of water and put them on top of a coin, and you'll actually see that the water extends past the edge of that coin. It's a property of water called cohesion. Cohesion, meaning that water molecules stick together. So they'll expand out on the side of the coin because the hydrogen in one atom or one molecule is attracted to the oxygen in another molecule. And this hydrogen is attracted to the oxygen in a different molecule. And this oxygen is attracted to some hydrogens that it sees in other molecules of water. So because I have areas, dipoles, with a partial positive and a partial negative, these two things are attracted to each other. That attraction is what we call a hydrogen bond, where the dipoles are attracted to each other. So hydrogen bonds form between positive and negative dipoles. Between positive and negative dipoles. That's an important idea about hydrogen bonds. Help me out in the chat. What do we want to know, or what do we need some more information about with these hydrogen bonds? I do see your question, Miriam, and I will answer that electronegativity question in a moment. I just want to see if we have any hydrogen bond questions. Okay, well, the chat has gone silent, so I'm going to assume we're still processing and catching it. Miriam had a question that related to our electronegativities, so let's go back here. She asked if electronegativity increases from left to right. That is generally correct, yes. The one exception to that is our little hydrogen over here. So hydrogen is more electronegative than the rest of its friends here on the left side. Hydrogen wants those electrons a little bit more than the rest of these ones do. But in general, things that are farther over to the right are more electronegative. Things that are farther to the left are less electronegative. So in general, that is true. Yep. Okay, so here's the last thing that I want to talk about then. We have talked about dipoles, so partial negative and partial positive, and we've talked about hydrogen bonds. The last thing I want to mention then is the difference between polar and non-polar things when they're interacting with water. So we said that water has partial positives and partial negatives. So that's kind of hard to see. Let's put it over here. Partial negative and partial positive on water. I should probably just draw them. That's pretty pathetic. Let me check my chat really fast. Do we have some specific atoms that can only attack the hydrogen to make a hydrogen bond? So hydrogen bonds typically form between oxygen and hydrogen or between nitrogen and hydrogen. So when we talk about, we'll go back here with these hydrogen bonds here, the picture that we're looking at right now shows oxygen and hydrogen being attracted to each other. We will also talk about, though, how in DNA, in your genetic information, we can also see nitrogen instead of oxygen, see nitrogen interact with hydrogen in that way. So generally speaking, it's just going to be oxygen plus hydrogen to be a hydrogen bond, or it's going to be nitrogen, that's an N, nitrogen, plus hydrogen, making that hydrogen bond. It is also possible that we could do things, sometimes sulfur can do that as well, but it always has to be something that's much more electronegative, that's being attracted to hydrogen's dipole. But these are the two most common ones. The ones we'll see in our class are oxygen and nitrogen making those hydrogen bonds. All right, so last idea today. We're looking at water, and we're looking at a compound here with dipoles. Hey, let's see who wrote it down and paid attention. I'm looking at a compound right here that has a positive dipole and a negative dipole. Which kind of chemical bond is holding these two things together? What kind of chemical bond is inside this molecule right here? Yes, lots of us are chiming in. This molecule has a polar covalent bond. It has a polar covalent bond because I have two dipoles. I make dipoles when I don't share electrons equally. Remember, covalent is sharing, polar is unequal. So this molecule has dipoles. It has partial positive and partial negative. When I put a molecule that has partial positive and partial negative in with water, remember that water also has partial, oops, trying to draw there, partial negative and partial positive. Partial negative and partial positive. Water also has polar covalent bonds because water has dipoles. So if I take water with its polar covalent bonds and I mix it with something else that has polar covalent bonds, my little compound that has those dipoles, has those polar covalent bonds, says to water, hey, I like your charge. You've got positive, you've got negative, I've got positive, I've got negative. Let's surround ourselves with each other. So in my picture up here, we've got our negative dipole and our positive dipole on our molecule that's in the water, the positive parts of this molecule are going to put themselves closer to the negatives of the waters that are around them. This negative part right here is going to surround itself by the positive parts of water. So a polar covalent compound, polar covalent bonds, which are in, we call them polar molecules. They're in polar molecules. Polar molecules surround themselves with water. Surround themselves with water. They put water on all sides of them because they have a partial negative and a partial positive, and water has partial positives and partial negatives. So polar molecules will surround themselves with water. Now, I need you guys to write this down because students often get this mixed up. Here's a really important idea right here. Polar compounds do not dissolve in water. They do not dissolve in water. I need to define this word dissolve for you so that you know what I mean when I say dissolve, because you'll see this on the homework and you'll see this on the exam. When I say the word dissolve, I mean that the chemical bond falls apart. Falls apart. We've got a partial positive and a partial negative in this molecule. But this molecule never stops sharing electrons between each other. These two atoms stay connected. They'll also be attracted to the water that's around them, but they're never going to leave each other. They stay attached to each other. Later in the lesson, so during our office hours tomorrow, we'll talk about what happens when you have a bond that we don't share electrons. When you put that kind of bond, an ionic bond in water, it will dissolve. It will completely fall apart. But a really important idea for us to keep in mind, because it always mixes my students up, polar compounds, they don't dissolve. They surround themselves by water. Absolutely, they love to have water around them, but they're not going to fall apart. They still stay together. They still stay intact. So polar compounds, because they have dipoles, will surround themselves with water. And polar compounds have polar covalent bonds. When we talk about something that does not have dipoles, things that do not have dipoles, they share their electrons equally. So equal sharing bonds, what were those ones called? Equal sharing. An equal sharing bond. Exactly. Non-polar covalent bonds. Non-polar covalent bonds. If you have non-polar covalent bonds inside of you, you are a non-polar compound. Non-polar compounds share their electrons equally. Because they share their electrons equally, they have no dipoles. No dipoles. No areas with partial positive or partial negative. So when water comes up to these things that have no partial positive and no partial negative, water has its negative charge over here, has its positive charge over here. Water has charges. It comes up close to these molecules that don't have charges, that are sharing their electrons equally. And these compounds here, these nonpolar compounds, say, I don't need that kind of negativity in my life. You got a partial negative charge. I don't need that. I'm happy. I have no charges. Or when the partial positive charges come over from the hydrogens, these molecules say, I don't need that kind of positivity in my life. That's too much. I'm happy and I'm stable. I need no charges. When we talk about nonpolar compounds completely stay away from water. So remember that polar compounds surrounded themselves in water. Nonpolar compounds push water away. They want nothing to do with water. There's my typo. Gotta love that. Push water away. Reminder that polar compounds surround themselves with water. So this is the idea we're going to end with today. A nonpolar compound, which means it has no dipoles, meaning we're sharing our electrons equally. If you share your electrons equally, you push water away. You don't have time for that kind of negativity in your life if you're a nonpolar compound. If you're a polar compound, you love those dipoles of water. You're going to surround yourself with water because you love to interact and form hydrogen bonds with its dipoles, with its partial positive and partial negative. So nonpolar compounds versus polar compounds. It has to do with the kinds of bonds that they have, chemical bonds that they have, and that will dictate the way that they interact with water. That's all I'm going to cover today. Help me out in the chat. What questions do we still have? Or give me an emoji. Nicole asks, so neither of these bonds dissolves. That is correct. Neither of these bonds dissolve. Remember that polar compounds, they still stay attached to each other, even though they interact with other molecules. But they're not gonna lose each other. Any other questions? That was a good one. Neither of these bonds dissolve. Draw you a little penguin. Too small. He's looking pretty terrible. There's my poor demented penguin. He's having some trouble today. All right, well, if we got no more questions, I'll go ahead and stop the recording for today, and I'll stick around for some time if you have any particular questions. We will pick up with the rest of the lesson tomorrow. If possible, try to work through the rest of lesson 3 between now and tomorrow's office hours at 9:30, so you have an idea of what questions you still have. And also try looking over the homework assignment too, just to see if there are particular types of questions that you feel like you don't understand well enough yet. So we will call that a day. I'm going to end that recording and I'll stick around for any last minute questions you have.